I will be talking about how I accelerate bioinformatic workloads, but we actually have work on various types of data sets, including large language models and other types of machine learning databases and so on. So for today, more about biowork that my team has done with Rob Knight, Nima Moshiri, and many students and postdocs at UCSD. Why bio? Well, data is exploding. If you look at genomics alone, you can see that we're now in tens of billions of human genomes sequenced. Data doubles every 7 months, and it has well surpassed Moore's Law a while ago. It is reaching exabyte scale and surpassing YouTube and X combined. On top of that, you have mass spectrometry, which is used for proteomics and metabolomics, on top of the normal stuff you do with mass spectrometry, all the other chemicals. So here at UCSD, we have one of the biggest repositories of proteome data sets. And the little screenshot I have here is totally out of date. I think we're closer to over 700 terabytes now. I can't keep up with it. The monthly data submissions to one of the databases increased by 100x continually. So you can see that the deluge of data is just crazy. So what are these omics disciplines? That these disciplines really play a significant role in developing personalized medicine and drugs that would be used for individuals. They consist of genomics, which is a study of a DNA, transcriptomics, which looks at RNA. RNA is sort of like DNA, one letter is different. Then proteomics, which is all the crazy proteins you have in your body and outside. Proteins really do a lot of the work, actually, inside of us. And metabolomics are the metabolites that consume and produce energy that we need. Now, if you look at how all this fits together, we take these different data sets and we want to integrate them in together with any data that we know about various kinds of diseases and individuals that have those diseases. And we want to use these data sets to make some decisions on what types of medicines should be developed. To do all this, you actually have to analyze an enormous amount of data. For genomics and transcriptomics, this really boils down to long strings of characters. For proteomics and metabolomics, they depend on mass spectrometry, which is essentially a really big bar graph. So from my perspective, both of these work really well if you have highly parallel hardware. They really do not work well on standard CPUs, which are designed for 64-bit numbers. And both of these data sets feed into downstream machine learning tools, which, again, are going to use large-scale data. So to accomplish personalized and secure drug discovery, one of the biggest pieces of the puzzle is the top left. So if you look at the overall path to get to the market, from an idea to actually medicine that you can buy, takes about 15 years. Almost half of that time is wasted on data analysis. And I say wasted because look what happens. We start from infinite possibilities of compounds. We identify 250 after nearly 7 years of analysis. Out of these 250, we start clinical trials. 90% fail. So we wasted 7 years in order to have only 10% of our bets potentially work. That is a huge waste of energy and resources. So one of the goals that my team, together with the center that I'm running, and I'll tell you more about later, has set out is to speed up this by at least 100x. We figure if you get at least 100x, that gets us in the region of months. So to do this, we're looking at all critical problems. We're looking at genomics, as described by short and long reads, proteomics and metabolomics, primarily through mass spectrometry, and then we'll combine these data sets to actually perform virtual drug screening. And let me kind of walk you through every one of these pieces that we have worked on over the last years. We first started on short reads as I started collaborating with Professor Rob Knight. And if you know Rob, you know that he's completely obsessed with bacteria. He's the Mr. Microbiome. So my first conversation with him was, can you speed up sequence alignment and classification of bacteria? He said, I think I can. If it's just a bunch of characters, I think I can figure this out. So that's how we got started. This was end of 2019. What happened very shortly after that, you guys know, COVID-19 hit and pandemic came. And at that point, we started looking at how can we actually speed up COVID-19 analysis. So if you look at the way the typical sequence goes, and this is regardless of whether you got human, bacteria, or virus, you're going to extract the sequences using one of the famous sequencers from a company like Illumina. From there, you're going to get lots of 200 to 300 long character strings that now you need to do something with. What you do is, step number one, you're going to trim the adapters. These adapters are placed on there to essentially label what it is and to make sure that you can run it correctly through the machine. Then you run through alignment. This alignment is very important for a couple of reasons. First of all, you want to get rid of anything that's human if you're looking for COVID-19. Secondly, you want to figure out what might be COVID-19 and not something else like bacteria or who knows what else. Okay. And the third piece is primer trimming, which is again a cleanup phase that has to do with the way you process data. This green box that you see here is the same regardless of what you're processing. Whether you do humans, bacteria, plants, doesn't matter. It all looks the same. What is different is what happens after that. Now, this step took many days in January 2020, like almost a month, actually. And in fact, to get it to run reasonably, we spent a lot of time optimizing the way this code was written and the way it was run. For example, what you don't want to do is put terabytes of data on a network disk in a cluster that runs a bunch of different applications, because you're going to spend most of your time waiting for data to come from that network disk. So there are lots of little optimizations that got us actually down to hours, more correctly, 18 hours. From beginning to end. After that, we need to do multiple sequence alignment and then inference and clustering that allows us to figure out exactly what particular strain of COVID-19 do we see, or what strains do we see. Now you can see that spending 18 hours is significantly better than a month, but still leaves much to be desired if you're waiting to figure out, am I sick or not sick? Especially, for example, if it's not COVID-19, if you're instead a newborn who may have a significant genetic disease that needs to be treated immediately. So to deal with this, we looked at what exists in the market. The best, fastest solution that Illumina was selling is Dragon FPGA-based local alignment tool. This one gives you about 7.5 times speed up over the state-of-the-art CPU-based code. What my group worked on is improvements on this. So we first changed the way we treated this text. We used a new way to represent the data, and along with new type of machine learning called hyperdimensional computing. This allowed us to speed up by about 200 times. And then the last step we did is we realized that FPGAs are great, but really when you talk about billions of characters that need to be aligned, it's much better to do this in memory or in storage instead of moving the data someplace else. So what we did is we actually designed an accelerator that uses non-volatile resistive RAM to do alignment directly inside the memory. So data never moves. And with this, we got about 2,000 times faster runtime. So first step, as I mentioned to you, was pre-alignment and post-alignment steps, adapter and primer trimming. Those two steps, turns out, are almost exactly the same. The only difference is what you actually feed into the engine. In the adapter trimming, you're going to feed in adapters and whatever commands, whatever you're looking for. And on the primer, you just need to feed in a list of primers and the options related to that. Everything else is the same. So we implemented this on an FPGA here at Nautilus. And we found that we were able to do adapter trimming, quality pruning, and fixing up to 30 times faster compared to the fastest tool that runs on 12 threads of CPU. So fairly significant improvement. On primer, we were 175 times faster. as compared to the state of the art with only 25% of FPGA resources. Basically, what this tells you is you could improve the speed much more. The next step is alignment. So that's kind of the middle of that green box that I was showing you. For the alignment, we went straight into in-memory acceleration. FPGAs actually would help as compared to CPU, for sure, but they cannot approach the speed that you want and that you can get if you actually never move data out of memory. So the idea here is that we would place whatever sequences we'd had in various locations in resistive RAM and then add a little bit of compute to every single location. Since alignment is really a dynamic programming type operation, we had to actually store the trace forward and trace back information in the local memory cells. Putting all of these pieces together, we were able to be about 250 times faster than the best FPGA implementation that Illumina sells for tens of thousands of dollars. And we could run, as I said, almost 2,000 times faster than CPU. And you can see kind of how we scale. We essentially have very nice scaling time regardless of what size of the problem we got. So putting all this together, you can see now that we can align, so go through the first box that is needed for COVID-19 and anything else that involves genomics or transcriptomics within about seconds. If we want to do the rest of the analysis for FPGA, you got to do multiple sequence alignment and phylogenetic inference and transmission clustering. So let's look at these pieces next. So to do transmission network analysis, you really are asking a question of who transmitted this virus to whom. In order to do this, you want to start with the rightmost side with our sequences. We figure out where those sequences fit on the evolutionary tree of COVID-19 in this example. Once we have that, then we can ask the transmission network. Once you have that answered, then you actually can inform people and their contacts. So to do this, we used FPGA-based accelerator to compute very quickly pairwise distances on nucleotides. And this was able to run within 4 seconds on full-scale data sets at 170 times less energy than running on a CPU. And you can see that pretty much across all the data sets, we actually have better execution time and better energy efficiency. Now, the last piece in this story is doing the clustering and classification. And for that, I'm going to show you how this works on bacteria instead of on COVID-19. So as you can see from this picture, the stuff right before, this first box is the same between bacteria and virus. The difference is down below. So the last piece that runs beta diversity analysis, that's basically asking what are all of the bacteria you have in there and how well represented are they? What is their frequency? That is done using Unifrac tool, which was accelerated by Rob's group on GPU by about 1000x. My group really focused on clustering and classification because those were the problems that had truly humongous data set sizes. So to do this, we took actually a twofold approach. First strategy was to recast the problem into new data representation, which is much better for highly parallel FPGAs and also in-memory processing. The second is obviously to implement the acceleration itself. So let me walk you through hyperdimensional computing and the data representation using hypervectors. In hyperdimensional computing, we take data, and here I have pretty pictures of cats. I would take each individual image and I would encode it into a hypervector that is 1,000 or more bits. I would do the same thing with the dogs. And to perform training, I would actually have to add these hypervectors together. So here's an example of how encoding might work. For a particular picture of a doggy, I would take for each pixel the location of the pixel and the color of the pixel. Those get combined into a single hypervector. You can see the equation on the bottom. Once I have that single hypervector, now I'm ready to do my training. Training is just addition of all of the hypervectors that represent each individual image. So all the hypervectors for cats get added together to create a cat hypervector. And here I'm showing essentially a binary distribution. where -1 is a 0 and +1 is just logic 1. Same thing for the dog hypervector. If I want to classify, I would take an unknown piece of data, I would encode it into hyperdimensional space, and now I have a query hypervector which I can compare. If it's binary, the comparison is super simple. It's just Hamming distance. If it's non-binary, then you need to do cosine similarity. To do clustering, you can do exactly the same thing in terms of encoding and in terms of searching for the closest match. The difference is that in order to cluster, we need to start with some cluster centroids, and we need to find what's close enough to each of the centroids. So here we have a loop that keeps essentially identifying better and better centroids and reclustering until we reach some error tolerance that we have specified. So you can see we're relatively easily, but now with very large data representation, we can do both standard classification and clustering. We then implemented this, and we have libraries available for CPU, GPU, FPGA, and we have actually designed accelerators, ASIC and processing in memory on resistive RAM. And you can see here that our inference runs as much as 3 to 5 orders of magnitude faster. On FPGA specifically, we're about almost 2,000 times faster than CPU for encoding and hundreds of times faster for the other steps. Similarly for clustering, you can see dramatic speedups if we use FPGA or if we accelerate in memory. This is again because we have this nice combination of an algorithm that's perfect for in and near memory computation because it uses very simple bitwise operations, and you have data that really does require this kind of highly parallel computation. So we implemented this for the bacterial classification, and here we trained hypervectors to represent each alphabet of DNA. So a DNA becomes essentially a sequence of those alphabets. And for classification, all we have to do is do a pattern match against the query to all of the stored sequences. The results were that our design that used in memory was almost 10,000 times faster. So really, really big gains. So now that you have seen what we can do with just standard genomics approaches in terms of classification, clustering, and also alignment, I would like to give you an idea of the types of problems that can be solved. So in this case, we're looking at human cancer variant detection. So again, the first box on top is exactly the same process that I described earlier. The box on the bottom is where the difference lies. So for this variant detection, the state-of-the-art was deep variant that was implemented by Google. Essentially what Google did is they said DNA is sort of like images. If you take DNA and assign a color to each letter and then put it together in a picture, you can use all of the same tools that they already had for object recognition in images, but instead now you're looking for patterns in DNA. That is what they implemented using GPUs. And you can see kind of how this stuff looks like when you actually plot the image on the bottom left. What we said is, let's see how well this would work on FPGA and CPU. And you can see that on FPGA and CPU, we get very, very comparable accuracy. The next step was to actually accelerate this even further than what FPGA can do. And for this, we again use hyperdimensional computing. You can see it on top right. What we did is we took a little piece of a already trained neural network. We froze that piece to act as our feature extractor, and then all of the training was done using very lightweight hypervectors. Remember that the training with hypervectors is just addition. There is no forward and backward propagation that's necessary. And then we implemented this and accelerated in hardware. And actually, we just got the chip back about a month ago. We're still testing the resistive RAM version of it. What we did find is that we can support at least 200 times higher throughput than GPU. And as I said, we're currently measuring on the actual chip exact details. So we were able to put together all of these different pieces using the same baseline. We provided an interface to Cheetah framework, which is what Rob's team and School of Medicine use for this kind of bioanalysis of the data. And we're able to show results for cancer variants, viral analysis, and microbiome analysis at the scale that runs hundreds to thousands of times faster than state-of-the-art. Where we are moving next, and what we have been doing in the last few years, is really looking at what do we do with proteomics and metabolomics. So proteomics, we actually took a very similar approach. We used UCSD's massive proteome database, and we used hyperdimensional computing again to encode the data from the mass spectrometry runs. The data from mass spectrometry looks like very simple bar graphs. Typically on x-axis, you have 10,000 to 100,000 positions, and on the y-axis, the range after normalization is from about 0 to 1,000 approximately. We profiled the whole workflow that currently existed in state-of-the-art tools and found that pre-processing was actually taking almost 60% of runtime. So that was actually the very first thing that we accelerated using near storage, meaning we had an FPGA that sat right next to SSD that allowed us to do this pre-processing extremely quickly. And then the last step is to actually encode the data into hypervectors for hyperdimensional computing and then store that data into SSD for further use for database search and clustering. So to accelerate both the database search, which is open modification search or OMS, and clustering, we actually implemented this on an FPGA. And as you can see, it was relatively complex, but the operations were dramatically simplified due to new representation that we use, because that new representation was doing only bitwise adds, multiplies, and only comparisons that had to do essentially with Hamming distance. It ran much faster than it would have if we had kept the standard representation that mass spectrometry needs. So the results that we saw were about almost 3,000 times better energy efficiency and about 40 times lower latency over other FPGA baseline that we compared to. So this is like better than the FPGA that already existed. We also took a step forward from there, and this is actually most recent work that was just presented a few months ago at a review that was hosted here at UCSD. This is collaboration across multiple universities and, as you can see, multiple companies. The idea was we wanted to see how much more can we push this kind of database search if we use in-memory and in-storage implementations. And when I say in-memory, I literally mean that compute is right next to your memory cell. And when I say in storage, I mean it's literally like right next to your storage cell. So our goal was to understand better for these new types of memories what is possible and maybe what doesn't work. And here we again leveraged the strength of hyperdimensional computing. We also collaborated with a professor at UIUC to develop a compiler that can take any general purpose application that uses hyperdimensional computing and map it onto CPU, GPU, FPGA, or any one of the accelerators you will see in a second. We looked at two different applications where I'm going to show you results on a small data set. Small data set was used so that we could actually run everything on the samples that we had. So for some of the chips, we only were able to run very small amount of data on an actual chip. And then we ran on a larger data set, which was 154 terabytes for massive. There we wanted to show the kind of benefit you would get if you had big enough chip and you built a system out of those chips. So here is the small data set result. On the left side, the stuff that's red was actually measured using that small data set. On the most furthest left is the state-of-the-art implementation, which actually uses GPU and CPU combination. All the results are normalized to that implementation. The very next column is our GPU implementation that uses hyperdimensional computing. So this shows you just the benefit of a different algorithm. And you can see here that we get improvement in speed of almost 40 times just by changing the algorithm. The next line over is what if we improve the GPU? So thanks to Nautilus, we're able to run on H100s. And we ended up with a speed up of about 100 times over what we started out with. And then the last red column is FPGA implementation. And here we have 172 times improvement. Everything that's in green on the right is measured on an actual device. But because many of these are brand new devices and implementations that don't exist at big enough scale, the rest of it is either emulated or simulated. So the first column is DRAM. Here, we're actually doing compute near high bandwidth memory. And you can see here that the speed up of that is on the order of about 100x, very similar to what you see with a good GPU, which is really not that surprising because good GPU has high bandwidth memory. The next two columns look at resistive RAM. This resistive RAM has two different technologies. 130 nanometers is what was actually produced at TSMC and what our collaborators at Stanford measured. The 40 nanometer is what we produced at TSMC and what we're measuring right now. And you can see that here we get almost 200x when we move to 40 nanometer. Now, the GPUs are on the order of about 4 to 7 nanometer. So there's a big difference here in the technology. The next column is a new type of phase change memory. So this has not been manufactured. This was just produced in small few bit sizes by my collaborator at Stanford. The nice thing about this phase change memory is that it has better properties for reads and writes than resistive RAM. And this is why you see about 1,200x improvement in speed. The two technologies on the far right are 3D NAND flash and 3D ferroelectric NAND flash. Today, what you have is standard NAND flash. The 3D is when you start integrating in the third dimension inside of the silicon. So that you cannot buy in the market quite yet. But we could actually measure the speed up of about 60x. On the right side is ferroelectric NAND. This is still in research. It was produced. You can see a picture of the device that was actually measured. And here you can see almost 1,000x improvement. So key takeaways are that generally you want to run this stuff in resistive RAM or phase change memory if you can, because it's much faster. If you can't, then ferroelectric NAND is obviously the best choice. Now, looking at the large-scale data, and this is now 154 terabytes of data, the assumptions were that we're able to fit 1.67 terabits per package, which is what you can buy in the market today, and that all of the packages were on the same printed circuit board. So if you needed more data, you basically had to go down the printed circuit board to get it. Key takeaways that you see here is that you would need 800 GPUs to fully fit the reference data set in GPU memory if you were using state-of-the-art, which is the far left. Instead, you could run it on our resistive RAM or phase change memory at a speed that's 200 times faster than state-of-the-art. And you can see also that for NAND flash, we can actually get pretty good scalability. So on the very far right, we're almost at 4,000x improvement in speed. Now notice I haven't been quoting energy. If I start looking at the bottom row, which says energy, then there is no competition. Running things in memory and storage will win by big order of magnitude difference. Okay. So far you have seen that what we've done for genomics, what we have done for proteomics and metabolomics. Where we are moving now is the piece where we select the most promising drugs. For that, we're actually collaborating with Rob Knight and Peter Dorstein at UCSD. Peter Dorstein is a world leader in metabolomics and has provided us access to metabolomic data sets that we need. Rob Knight is doing the genomic piece. They're jointly running a big Alzheimer's study, and we're able to get the data from them and show that, in fact, we can combine the data analysis from genomics and metabolomics and predict actually better than the state-of-the-art the outcome of drug influence on the development of Alzheimer's in these patients. This work has not been published yet. It's actually in process. So I'm not showing any results as a result. So next time, hopefully I see you, I will have some better news. The reason why we can do this is because we have amazing support from people like you. We use your infrastructure all the time, and we have great support from Jump 2.0 Center. This is a center that I'm running, Prism, and that actually Tom is helping me run. And we got a whole bunch of faculty involved in this work. Specifically, the center consists of 24 PIs across 13 universities nationwide and about 230 PhD students. Thank you, everybody.